Good morning, all of you. My name is Dr. Neha Hamilton, and we were studying research methods and management, and we have already completed the methods of research. We have completed purpose of research. We have completed types of research. Uh, we have also completed the concept of sampling, but the topic which is left is basically the specific types of random and non-random sampling. So today we are going to understand in detail that what are the specific processes of sampling in a particular population. For example, you are doing a research in a particular discipline, right? And it depends on the type of the topic that you take, on the kind of sample you select, right? So we are going to understand that what are the various kinds of probability and non-probability sampling techniques that you can use in your respective research studies. So let me share the slide. I hope the slide is visible to all of us. And basically, in sampling is taking a representative population, representative part of population from your population that is the right representation of your population. So that is called as sampling, right? So I assume that we have covered the concept, the basic concept of sampling. For example, in this circle, this big circle, this shows the total population, and this small red rectangle that you are seeing shows the sample that you have chosen from the population. So, sampling is a process of selecting observations to provide adequate description and inferences of the population. It is a unit that is selected from population and represents the whole population. The, what is the purpose to draw the inference? See, we are selecting this sample as a representative of the whole population. So, what we are going to do is we are going to generalize this study to whole of the population, right? So that is called as sampling process. Now this is a diagram that we have discussed. Let's do a quick revision of this diagram. This purple circle actually represents a population, and this blue color of rectangle that you can see inside is the sampling frame. Now through a sampling process, you select this population. From the population, you select this sample, and basically you do all your research studies on this particular sample, and whatever the inferences which are generated from this sample are applied to the whole population. For example, vaccination trials have been done in very specific population, and on the basis of those trials, on the basis of results of those trials, we are implementing the result to whole of the population, right? So that is called as sampling process. And why census inquiry is not possible all the time? Because census inquiry means we are taking each and every individual of the population in the in the research process, which is actually not possible. Now, why this is not possible? Because it takes a lot of time, it takes a lot of resources, it takes a lot of money, financial resources also, if you want to cover all of the population. For example, you want to do a study on Indian population. So Indian population is like 130 crore people. So you cannot go and reach to each and every individual and do a particular research. You have to uh, you have to approach specific people to with those people who can give you a correct result. So that is the right approach of doing a particular research. It saves your time, it saves your resources, it saves your money, it saves your energy. So all these things you can invest in doing a proper research in the small sample size so that you get the right results. If you get the right results, then definitely if you generalize these results in the population, that is going to uh, help develop the population or help develop the society as a whole, right? So this is the sampling process. After sampling process, we have seen that there are two kinds of population. The first is called as homogeneous population, and the second is called as heterogeneous population. Right? Homogeneous population means all the individuals in the populations are exactly similar. There is no differences in them. Right? They are bilkul similar. Right? No changes whatsoever in the mental setup, in the opinion, in the physical characteristics, in the demographic characteristics. So, like, there is no difference in the population, right? So, that is called as homogeneous population. When we have a homogeneous population, I think random sampling is very good option because all the members of the population, they are similar. So you can take randomly any individual as a sample because everybody is similar. There is no difference, right? So that is called as homogeneous. Homo means same, genus means human beings, or a kind of a living race, that is called as genus. So homogeneous means the individuals which are very similar. Okay. Now let's move to the next slide. Now this is an example of heterogeneous population. That means individuals are different in they have difference in opinions. As you can see, there are two levels also. Three people are there at the back and rest of the people are there in the front. So when we talk about heterogeneous population, hetero means differences, different varieties, and genus means population, right? So whenever heterogeneous population has different variety of people and we need to take sample from that population, you have to pay extra attention. Why you have to pay extra attention? Because uh, every individual is different and you need to make a sample which is a correct representation of your population, right? So that's how heterogeneous population is a bit tricky. And in practical life, whenever you will go for research, you are going to find heterogeneous population. Why you are going to find heterogeneous population? Because most of the times people are different, we all are different. If two people have similar height, they may have similar opinion. If they have similar opinion, they may have different decision making process. They may have different hair color, they may have different eye color, they may have different ethnicity, right? So there are differences in people that we need to consider specifically when we are doing this sampling process. Now, this is the exact process of sampling that I was talking about. You have to define population, then you have to define the sampling frame, then you have to determine the sampling procedure. Now, what is this sampling procedure? Sampling procedure can be basically of two types. The first is called as probability sampling, and the second is called as non-probability sampling. Okay. Now, what is probability sampling? Probability sampling is basically a sampling in which we are taking the sample from the population on the basis of pure chance. So every individual in the population has equal chance of getting selected for a sample from a population in probability sampling, right? So it is kind of a random sampling uh, in which we are taking people on the basis of probability or on the basis of pure chance. So that is called as probability sampling. When we talk about non-probability sampling, see, non-probability sampling is different. How non-probability sampling is different? Because in non-probability sampling, basically, we are talking about researchers' uh, wisdom. We are talking about researchers' way of thinking. It can be different. If I'm doing a particular research, I may use a different kind of sampling method. If somebody else is using a different research, they may use a different kind of sampling method or sampling procedure, right? So that is called as non-probability sampling. When it depends on the researcher, then it becomes non-probability sampling. And when it is based purely on chance or probability, it becomes probability sampling. After you decide the sampling procedure, the next method is to determine the appropriate sample size. Now, how to determine the appropriate sample size? You can determine the appropriate sample size on the basis of your total population. We have different formulas through which you can understand or you can find out the appropriate sample size. But generally, if you are doing a thesis research, we always recommend you to take the sample size which is higher than 500 respondents, because higher the sample size, more accuracy the results, more towards more accuracy your research will go, right? More the sample means more the people are involved, more people are involved, more different kind of different dimensions of opinion you will receive, and the results will be better, right? So that's why we say that it is always better to take a larger sample. And if you are doing a thesis research, it is better to take a sample size of minimum of 500 respondents. Then you have to execute the sampling design. Okay. Now we have probability sampling and non-probability sampling. What is written, see in the slide? Probability sampling is based on concept of random selection, whereas non-probability sampling is based on non-sampling, right? So as simple as that. In probability sampling, the concept of random selection. Random means just we are doing the selection on the basis of anything, that is random selection. And what is non-probability sampling? We are doing the selection on the basis of uh, researchers' wisdom, researchers' researchers' opinion, or researchers' decision-making process, right? So that is probability and non-probability sampling. Okay. Now let's move to the specific type of probability sampling. In probability sampling, we have studied simple random sampling. In simple random sampling, you can see that this is like a telephone directory that you are seeing on your slide right now. If this is a telephone directory and the red circles are there are the part of sample that we are going to choose, right? So is there any similarity in the sample? I don't see any similarity in the sample. The numbers are not neither even nor odd, and also, when we talk about alphabets, these alphabets are also different, right? There is no similarity, no pattern, no sequence in this sampling. So that is called as simple random sampling. Okay, without any criteria, without any basis, when you take your sample from the population, that is called as simple random sampling. Now, what are the advantages of simple random sampling? The first is minimal knowledge of population is needed and easy to analyze data. See, wherever you are randomly selected the it is generally used in homogeneous kind of a population where generally all of the people in the population are similar, right? So when minimum knowledge of population is needed because we are selecting the sample randomly, right? There is no basis on the basis of which we are selecting the sample. So minimum knowledge of population is needed, and it is very easy to analyze data because we are randomly selecting the sample from the population. Now, what are the disadvantages? Low frequency of use, does not use researchers' expertise, and larger risk of random error. See why there is low frequency of use? Because as I've already told you, we have discussed earlier that homogeneous population does not exist, right? There is no homogeneous population practically. Always exceptions are always there, but homogeneous population doesn't exist. And I already told you that random sampling is best when we are talking about a homogeneous population. So that's why the frequency is very less, right? And this is the biggest disadvantage of simple random sampling. It doesn't use researchers' expertise, right? So it doesn't use researchers' expertise means uh, as a researcher, my opinion, my wisdom, my knowledge has not been used. Why it's not been used? Because basically, I am taking the sample randomly from the population, right? I am not using any kind of technique. I'm not using any kind of system or method or hierarchy in taking this sample from the population, right? So I think you are clear with what are the disadvantages low frequency of use, and doesn't use researchers' expertise, and larger risk of random error. So random error can occur because if at all there are some differences in the population, if your population is a heterogeneous population, it becomes very difficult for you to cover each and every variety of the individual unless and until it is a very what do you say? It is non-probability sampling. You have to use non-probability sampling when you want to cover each and every part of the population. Now let's move forward. Now let's talk about stratified random sampling. Okay, so what is stratified random sampling? Stratified random sampling is basically a type of random sampling in which population is divided into two or more groups, which are called as strata. Now, what is the meaning of this word strata? Strata means you are dividing the population into small groups. Now, these groups have some similarity in them, right? That's why they are called as strata. Okay. For example, here the strata is on the basis of some kind of similarity. For example, here we have ethnicity, right? So here you can see Caucasians, you can see African Americans, you can see Hispanic Americans. So these are called as stratas. Stratas are the groups which are homogeneous in themselves, but they divide the heterogeneous population into homogeneous subparts. For example, we have the group which carries a mixture of different kind of people with different ethnicity, right? So what is strata? In strata, basically, you divide this heterogeneous group into homogeneous subparts, and these homogeneous subparts have some similarity in them, right? So here we are dividing on the basis of ethnicity. Okay. Now, what are the advantages of simple random sampling? Assures representation of all groups in the sample population. Characteristics of each strata can be estimated and comparisons can be made. So now we are moving from simple random sampling to stratified random sampling, right? So when we make strata, that means we are devising a way through which we can cover each and every variety or diversity in the population. I hope you are understanding my point. What I'm trying to explain is, for example, we have a population which has mix of uh, three kind of IQs: the people with higher IQs, people with average IQ, and people with lower IQ, right? So what will I do? I will try to compare people from all these three categories, but for this, I have to make three categories. I have to take an exam or test on their IQ to judge their IQ. Once I got the results of that test, I can differentiate into higher IQ people, average IQ people, and lower IQ people, right? And then, I can take a sample from the first group, then second group, then third group, right? So this is how it goes. This is stratified random sampling when you make homogeneous subgroups and you take samples from these subgroups. So that is called as stratified random sampling. Okay. Now let's talk about another type of probability sampling, and the example of this is called as cluster sampling. Now, when we talk about cluster sampling, see, before understanding cluster sampling, we have to understand the meaning of clusters, right? What are clusters? So, the population is divided into subgroups or clusters like families. So, clusters are generally on the basis of geographical areas or geographical boundaries. For example, if you are doing a research on Jaipur city, then you can divide the Jaipur cluster in Jaipur into various clusters. For example, one cluster will be of Mansarovar, one cluster will be of Jagatpura, one cluster will be of Sea Scheme, then one cluster will be of Vaishali Nagar. So these are different areas of Jaipur, right? Civil lines. So so you have to create clusters on the basis of geographical boundaries in a particular division, because geographical areas also decide the kind of people who are living in, right? The people who are living in Mansarovar will be facing different kind of issues. The people who are living in Sikkim, they will be having different kind of issues, right? So this is called as cluster sampling. So you are dividing the whole population into small clusters, and when you divide the population into clusters, you need to take a small set of data from this cluster, right? So that is called as cluster sampling. For example, here you can see section 1, section 2, section 3, section 4, section 5. Now, how I'm going to take samples? I am randomly selecting the samples from these clusters to make sure that almost each and every cluster is being covered. So this is called as cluster sampling. Now, cluster sampling is type of probability sampling. Now let us understand what are the advantages and what are the disadvantages of cluster sampling. Advantages means it can estimate characteristics of both cluster and population, and disadvantages is the cost to reach an element to sample is very high. Each stage in cluster sampling introduces sampling error. The more stages there are, the more error that tends to be. So, what are the advantages of cluster sampling? It can estimate characteristics of both cluster and population. So, here also we are dividing the population into clusters to make sure that the population has each and every kind of individual. Now, the disadvantage is definitely the cost becomes high, right? If I'm doing a research on Jaipur city, and if I'm residing in C scheme, and if I take randomly some 50 people from C scheme, it is very cost effective for me. But at the same time, one day I have to travel to Vaishali Nagar and then take 10 people from there, and then I have to travel someday to Mansoor and C scheme. So it is definitely cost consuming. Each stage in cluster sampling introduces sampling error. The more stages there are, the more error there tends to be. So these are the disadvantages of cluster sampling. I hope cluster sampling is clear, right? Next, we are going to understand systematic random sampling. Now, what is the systematic random sampling? In systematic random sampling, what we do basically is we order all units in the sampling frame. So we make a sampling frame, and we uh, make a sequence, and we set the population on the basis of that sequence. Now, what do we do? The first step is we make a sequence, right? Now, what is the second step? In the second step, we select every nth number in a particular list. Now, here the nth number is, if you see very carefully in the slide, the nth number is the third person. Third person means I selected first, second, third. Then I selected fourth, fifth, sixth. Then I selected seventh, eighth, ninth individual, right? So this is how the pattern goes. I'm selecting every third individual. For example, a research study on the class of MBA first semester. So I will set the individual in the increasing order of their roll numbers. Now I'm selecting every second individual, every alternative individual, right? So I will set a pattern, and accordingly in that pattern only I will select the students, right? So that is called as systematic random sampling, right? Now this n is called a sampling interval. Here the sampling interval is the third individual, but it can be anything. It depends on you, on the kind of sample you want, the size of sample you want. It can be 2, 3, 10, 15, 100, 150, 1,000, whatever you want. Depends on the size of your population and the size of sample you want to do, right? So here also it is kind of a random sampling, but we want to cover uh, each and every variety of individual by by taking the nth number in a particular population. Now let us understand the advantages of systematic random sampling. What are the advantages? The first advantage is moderate cost and moderate usage, simple to draw sample, and easy to verify. So, what is the basic advantage of random sampling? That the cost is moderate, right? Cost is moderate because we have divided the population into a systematized plan, and we are systematically taking the samples. So we have clarity in our mind, so it is a moderate cost. It will cost you moderately. Next is simple to draw sample and easy to verify. Easy to verify means you can go back to the population and you can reassure yourself that you have taken the right sample, right? Because when you set a system or when you set a hierarchy, you have made a plan, so it becomes easy for you to go about a particular change. Now what are the disadvantages? Periodic ordering is required. How periodic ordering is required? That means okay, so the thing is that it is not needed. Periodic ordering is required in stratified random sampling. Now let's talk about multi-stage sampling. Now when we talk about multi-stage sampling, it is carried out in stages, right? Using smaller and smaller sampling units at each stage. So how do we make smaller and smaller sampling units? See, as we have discussed that more the number of sample respondents you have, higher the accuracy of your research results become, right? So for example, here if you can see that we have primary clusters and then we have secondary clusters. So what are these secondary clusters? These secondary clusters are further subdividation of the primary clusters, right? Further subdividation means the areas where the clusters are big or huge, we have further subdivided these clusters into smaller clusters. Okay. Now, what do we do in simple random sampling? In simple random sampling, what we do is we randomly select uh, samples from the clusters as the primary clusters and from the secondary clusters, because this is random sampling, right? We are selecting the sample randomly. So how do we select it? We will randomly take it from the primary clusters and we will randomly take it from the secondary clusters, and then we compile it, and this will be the total sample size. So this is called as multi-stage sampling. Okay. Now, what are the advantages of multi-stage sampling? The first advantage is that it is more accurate and it is more effective. Definitely, how many levels, different levels, greater the levels you will make, greater the accuracy of your research results. That means if you are making 2 levels, the accuracy is high. If you are making 3 levels, the accuracy is higher. If you are making 7 levels, the accuracy is highest, right? So more the levels in your sampling, more the accuracy of your results. It is more accurate and it is more effective. Now, what are the disadvantages? Disadvantage is that it is costly. Okay, the first disadvantage. Next is each stage in sampling introduces sampling error. The more stages there are, the more error there tends to be. So we have to understand the concept of sampling error first, right? So what is sampling error? Sampling error is basically a error which happens at each level of sampling. So higher the levels, higher the sampling error can occur, right? Because even whenever we measure something from the machine, we take some chances of error. So we humans are doing this kind of a research. As a human, we can do error, we do make mistakes. So there is always a scope of error, and the scope of error increases if you broaden your criteria of taking samples. Broaden your criteria is what I meant is you increase the level of your sampling, right? So let's go to the next level, which is called as non-probability sample. And let us first understand what does this say, non-probability samples. The probability of each case being selected from the population is known. Units of sample are chosen on the basis of personal judgment or convenience, right? So, what is non-probability sampling? As I've already discussed, that in non-probability sampling, it whole soul depends on the researcher, right? If you are doing a researcher, it will depend on you. That what do you think, in your opinion, what is the best method of taking sample from the population, right? So that will become non-probability sampling. Because here you are not randomly selecting the samples. Here you are selecting the samples on the basis of a particular judgment or a particular opinion or a particular system, right? So that becomes non-probability sampling. Now let us understand what are the different kind of non-probability sampling. So the first technique is called as convenience sampling. What is convenience sampling? See, I understand that there are different kind of sampling techniques, but if you can relate the meaning of that sampling technique with the name can will only tell you the answer. When I say convenience, what is the meaning of convenience? Your comfort. Whose comfort? Researchers' comfort, right? So convenience sampling involves choosing respondents at the convenience of the researcher. So what I'm doing is I am selecting the sample on the basis of my convenience. As the name suggests, this involves collecting a sample from somewhere convenient to you, the mall, the local school, the church, and sometimes called accidental sampling, opportunity sampling, or grab sampling, right? So whenever I am collecting data on the basis of my convenience, right? For example, I'm living in, yeah, a particular area. I know this area very well, and I know the respondents very well. So it becomes easy for me to talk to the respondents to get data from them. So that becomes convenience sampling, right? So I will take data from the place from where I am living. So that becomes convenient sampling. Next is called as quota sampling. Now, what is quota sampling? The population is first segmented into mutually exclusive subgroups, just as in stratified sampling. Quota sampling is where the groups in the samples are proportional to the groups in the population. See, uh, what do we do in quota sampling? We divide the population into certain subgroups or certain quotas, as it happens in whenever you give a paper for government services. So what do? They have a quota system. That's what quota sampling is all about. They have a quota system so that uh, each part of the society should be covered. If we are covering the general candidates, we should cover the backward candidates also, the OBC, the ST candidate, the tribal area candidates. So they have made a system in which all the variety of the candidates who are living in the society, who are part of the society, should be covered. So that's why this quota method has been adopted in India. So same thing happens in research. The population is segmented into mutually exclusive subgroups, which are called as stratas, and from that stratas, these are mutually exclusive. When I say mutually exclusive means a general candidate cannot be a ST candidate, a ST candidate cannot be a ST candidate, a OBC candidate cannot be a ST candidate. If you have a specific type of religion, for example, you belong to minority, so you cannot. If you are a Christian, you cannot become a Muslim. If you are a Muslim, you cannot become a Sikh. So these groups that we make are mutually exclusive. There is no merging area in these groups, right? So that is called as quota sampling. Next is called as judgmental sampling. Now, what is judgmental sampling? Researchers actually employs his or her expert or judgment, right? So it is your judgment on what kind of technique, sampling technique you use. Maybe you devise a new method of sampling in which you are covering a large number of individuals, right? So in judgmental sampling, basically, you the researcher employs his or her own expert or judgment about a particular sampling technique. Now, what are the advantages? That there is a assurance of quality response and it meet the specific objectives. Disadvantages means biased selection of sample may occur or time consuming process. So it is a time consuming process definitely, right? And biased selection can occur. That means if a researcher is convenient or comfortable with a particular set of respondents, bias can occur and he cannot take the sample from the population on equally or on a equal basis. Next, we have snowball sampling. Now, what is this snowball sampling? The researcher starts with a key person and introduce the next one to become a chain. This is used when the respondents are hard to find. See, let me give you an example. For example, uh, you are doing a research on terrorists who are involved in attacking India, right? So you are studying or researching on an organization called Jaish-e-Mohammed. So you cannot go them, you cannot approach them, and you cannot talk to them directly. Why you cannot talk to them directly? Because it is not advisable to talk to people like this directly, right? So what will you do? You will basically search a person who can contact them, maybe a news reporter, a journalist, or maybe a political activist. You can choose a person like this who can take data for you. So this person is going to go with your questionnaire or your set of interview questions, going to ask to the terrorist and get you the data. So that is called a snowball sampling. I'll give you one another example. For example, uh, you are studying something which is in uh, you know, southern pole of the earth. You are doing a scientific study on the kind of the behavioral changes people are having who are living in the south pole or the north pole. So can you go to south pole or north pole? It is very difficult for you to do, right? But what if you can contact a psychologist or psychiatrist or a researcher who is actually residing there, right? You can send your research instrument to that person. He can facilitate data collection for you, and from there you can take the data, and from there you can continue your research process. So this is how it goes in snowball sampling, right? So that's it uh, uh, for today from my side. And now we are going to discuss some MCQs relating to these topics. Why we are studying MCQs, I have already explained you that MCQs clear your doubt, and MCQs actually give you a clear idea of whether you have clarified your doubts or not clarified your doubts. Okay, so I'm sharing the screen and we will very quickly solve some MCQ questions which are related to sampling. Okay, so the first question is research is ABCD. Research refers to systematic method of enunciating the problem, formulating a hypothesis, collecting data, analysis, and reaching conclusions. So definitely the answer will be A because we know what research is, right? It is not only hypothesis, it is not only collecting data, it is not only literature review. Next question is research involves. So what will be the answer? Not only inductive and deductive reasoning, but it involves inductive reasoning, deductive reasoning, hypothesis, data collection, everything. Now, what is inductive reasoning? Inductive reasoning is inductive means developing a theory. So definitely the answer will be A. basic difference between inductive and deductive reasoning that we need to understand. That inductive reasoning means the reasoning where actually what happens is that we we come out with a new kind of a theory. We enunciate a new theory or a new kind of a chapter. But what do we do in deductive reasoning? In deductive reasoning, we test a theory which is already existing, right? That's what we do in deductive reasoning. Now let's go to the next slide. Next question. Inductive reasoning moves from broad generalization to specific observations, writing paper, writing theory, or specific observations to broad generalization. So the right answer will be specific observations to broad generalizations, right? Because whenever we are making a new theory, writing a new theory, we create a broad generalization because we have just started with a particular research. Okay, which of the following are characteristics of the research? Accuracy, inductive reasoning, analytical, accuracy, inductive reasoning, and analytical or. So the answer will be or. We'll take one more question and then we'll wrap up the class. Basic research is used to enhance the basic research, analyzing the issue, solving the issues, solving real life problems or entertainment. So what is the answer? It is enhancing the basic knowledge. As the name suggests, basic research. So what do basic research do? It actually solves your basic issues or basic problems which are related to any discipline, right? So today, what all topics we have covered? We have covered sampling techniques, right? What is sampling? That you need to understand. What are the various sampling techniques that you need to understand? We have studied two different kind of sampling techniques. The first is probability sampling and the another one is non-probability sampling. So these two techniques you need to clarify in your mind. As a researcher, it is your responsibility to use the right kind of sampling technique so that you get the accurate results, right? It is very important to use the right kind of sampling technique. So that's all from my side. Okay, we have completed the sampling topic today. We have covered all aspects of sampling. We have covered probability sampling techniques. We have covered non-probability sampling techniques. We have covered all types of sampling techniques, right? Now I'm giving you a small assignment. Now, what this assignment is that you have to study 5 research papers, and from those 5 research papers, you have to find out what kind of sampling techniques the researcher is using and why. So, you have to make a table. In that table, you have to write serial number, then you have to write the topic of the research paper or the problem statement, and then you have to write the sampling technique that has been used in that particular research problem. This kind of exercise will help you understand that what kind of sampling can be used in what kind of research problem. So that's it from my side. Thank you so much for listening and joining. I am ending the meeting now.